Welcome to Lab 3 for Biology 102. My name is Mr. Donley. Today we're going to be going through and we're going to be looking at plant structure and soil salinity. Those are our major two topics today. So let's go ahead and get started. So you've already looked at plants generally. Now let's start taking a little bit deeper look, and more specifically we're going to take a look at the angiosperms. The angiosperms are the flowering plants. Okay. You've got different categories of plants. These different categories are grouped together either mainly by their characteristics. So in this case, angiosperms are grouped together because they produce flowers and thereby produce fruit. The gymnosperms are your cone-producing naked seed plants. These are going to be your pines. Okay. You also have your non-seed bearing mosses, ferns, and algae. But like I said, today we're focusing primarily on the angiosperms. Alright, so to produce complex structures, plants need cells that are organized to function together for a common goal. In animals, humans, we call these tissues. They're the same in plants. So in plants you have dermal, vascular, and ground tissues. And ground tissues can then be subdivided into parenchyma, collenchyma, and sclerenchyma cell types. So, well, what does all this mean? Well, we need to figure out within an angiosperm, is it a monocot or a eudicot? Okay, how do we do that? Well, there are certain characteristics, like I said earlier, that we're going to use to identify or separate these two into different categories. Why do we separate all these into different categories? The big reason why we separate all these into categories is to help us figure out lineage. What came from what? What ended up actually giving us this one specific species? How did we get here? Okay, so grouping all these and categorizing these together, we create things called field guides. These field guides are keys we use to actually figure things out. Well, we have to have these characteristics in these groups to help us build these keys and guides. And that's how we figure out what plant we're looking at it, instead of just walking out there and starting to name new plants because, well, it feels good. So the characteristics that differentiate monocots from eudicots, okay, the old term for eudicots is dicots. It's really, the main one is how many cotyledons. So in this case, you see for a monocot there's one cotyledon. For a eudicot there's two cotyledons. So when it actually emerges from the soil coming from a seed, how many leaves does it have? If it has one, it's a monocot. If it has two, it's a dicot or a eudicot now. Okay? So another way, because you can't always be there when they actually push through the soil, so you have leaf veins. If you look at the actual leaves themselves, you'll see that with your monocots, they're going to have parallel leaf veins. And with your eudicots, they're going to usually have these branched veins. We'll see a couple examples of those today. Now, in your stems, if you go through and take a cross section or you cut a piece of the stem out, you're going to see that the vascular tissue or the tissue inside of it that we talked about earlier, they're going to actually be arranged in different patterns. Okay, so in the case of a monocot, you've got these vascular bundles that are going to be scattered. Okay, so they're scattered in complex arrangements. Now, if you look at the eudicots, these vascular bundles are going to be arranged in a ring. Okay, that is in the stem. Now, keep in mind there's going to be a difference between what you see in the stem and what you see in the root. Okay, we'll get to that here in a minute. So, for the flowers, in this case, if you just so happen to be out in the spring and you're looking at this plant, you can also tell the difference between monocots and eudicots by the number of flower parts. They're either going to be in multiples of 3 or multiples of 4 and 5. Okay, there's a couple of examples today. Then you've also got your fibrous root system. If you actually uproot the plant, you'll see that monocots will have this fibrous root. You usually see this if you've ever been out weeding and you pull up the grasses that you don't want. Okay? If you happen to pull up a young tree or anything like that because it's in your yard, you'll find that there's this taproot structure if it is a Eudicot. And you see this main root right here. That's the taproot. Okay? Alright. So let's look at an example. So in your lab, under part 2, you've got a table under procedure. These are the images that you're going to use to fill this in. So the first image here is a buttercup root. Okay, so we know we're looking at the root. So if we're looking at the root, what we saw earlier isn't going to make a lot of sense on that table. So if you look in your lab manual, you'll actually see that there's, on the far right hand side of their table in the lab manual, another characteristic. In this case, it shows that the arrangement of the primary vascular bundles in the root for a monocot is going to be in a ring. Okay? But in a Eudicot, it makes this interesting little X pattern. This X pattern is what tells you automatically that it's a Eudicot. Okay? So I'm going to go through the rest of these so that way you can see them and have them for your lab. So in this case, you're looking for a specific characteristic. What do you see? If you notice, there's some parallel veins. Those parallel veins are going to be telling you it's a what? It's actually going to be a monocot. Okay? Now we go to image 3, viburnum. Take a minute and look at it. What do you see that's unique about this leaf? What is different from the previous leaf that you just saw? In this case, note that it's branching. Okay? See all these little branches in here. That is a huge advantage because that means that you can actually, as a plant, they can take and spread out those nutrients and get all of that energy that is collecting in the form of sugars back into the plant easier. You basically created more highways. Alright. So in this case, now we see a flower. How many petals does it have? This is going to tell you. Okay? I'm going to leave that one up to you. Another flower. Trillium. Look at the number of flower parts. How many does it have? Which one is it? A monocot or a Eudicot? Okay. So, now I've got some bonus ones. These are from my house. So, with this, I found a flower, and this is from a tulip tree. Is this one a monocot or a Eudicot? Take a minute and count the flower petals. You see that it actually has 1, 2, 3, 4, 5, 6 different flower petals. Oh, you also get a bonus. You get the leaf. Notice that it's, branching. So that tells you it's a Eudicot. This is commonly referred to as the yellow poplar tree, more commonly the tulip tree. This is a native species. One of the ones that you can actually identify with the keys that I'm going to give you with this lab. Okay, so now we're looking at a root structure. This is something that I pulled out of my yard. I didn't want it in my yard anymore. So when you look at this, it kind of looks like it might be fibrous, but notice there's one that's just this huge root right here. Oh, okay. Well, there's your taproot. So if it has a taproot, I want you to go back, look at your table, and see which one this is, monocot or Eudicot. Okay, now we've got another common native species, and these trees are huge. They're really, really cool. They're annoying if you walk barefoot, but this is the American beech tree. So the American beech tree, if you notice, it's going to also have these branching, but they're so uniform. you've got these branching veins in the leaf, this vascular tissue that branches. So in this case, it's got to be a Eudicot. And now we get to the arch nemesis for me. This is something that you really want to know how to identify, especially if you're going to go out in the woods. So in this case, this plant is poison ivy. How do you know? Well, for one, it's got a glossy, waxy coat, this waxy cuticle on the leaf. Okay, this waxy cuticle, you can see the shine right here. Okay, you also look at the leaf and notice that there's these little indentations. There's a little indentation right here you can't see, and they have two of these indentations, and they have a pointed leaf. Okay, and the younger leaves, these are both from the same plant, you can see it a lot clearer. Now when you get into these bigger, broader leaves, it's hard to really see where in the world that little indentation is. It's right here, it's right here. Okay, so it's a little more clear on this leaf right here. So this is actually growing up to about 30 feet on this tree. This tree that it's actually growing on is a yellow poplar, the tulip tree, and I did it the favor of removing this poison ivy the other day. Okay, so this one can cause you some long-term rashes if you actually come in contact with it. It's a urushiol. Okay, this oil is going to cause you problems by creating a rash if you're allergic to it. Not everybody's allergic to it. Alright, and so in addition to what you have in your lab manual, I've added one more section. And this is going to be something that you're going to see on the actual lab report. This is your field identification guides. I've given you two of them from the Virginia Forestry Service. So I would like for you to take and go outside into your yard and identify two different plants. You can choose tree, native shrubs, woody vines. You'll be taking pictures of these, and you'll be taking pictures that show the characteristics that you use to identify it. Okay, this is probably going to be the longest part of your lab, but there's some help out there. Okay, some of it I've given to you in this lab. You can use some of the species that I have here. Just please don't touch the poison oak, or excuse me, poison ivy. So now we're going to move on to part 3, the root system. For this lab, for part 3, you've got some different regions that you want to be able to identify in a slide. So in this case, you want to be able to notice the actual root cap region. The root cap region is this region right here. This is what allows it to push through the soil, because the soil doesn't want to move out of its way, so it has to have something that it can use to wedge through the soil. Okay? Then, to give it that force, to give it that pushing force, you've got the zone of cell division. This zone of cell division includes what's called the apical meristem. This is the region where you are actually taking and going through active mitosis of the cells. The active mitosis of the cells means they're reproducing themselves, and then after they've reproduced, which you can identify in a slide by the number of times you can see mitosis happening in cells that have stopped. Okay, once they've actually multiplied, then they're going to go and elongate. Okay, they start to elongate, and that's what's giving that little bit of extra force to push that root cap into the ground. After it elongates, you've got the zone of differentiation. This is where the cells start to have different functions. They start to do slightly different things. And this is the really cool part, because at this point you now have cells that are now figuring out what they want to be when they grow up. Okay, so they can end up becoming root hairs, or they can end up becoming cuticle cells. They can start to, cuticle manufacturing cells. Okay, but in this case you can have a couple different functions. Okay, Now let's look at a real slide. So in this case, this is from Allium cepa. Allium cepa at 40 times magnification. So we're going to zoom into this little region that you see here, and notice that there's a lot of little squiggly things. What are those squiggly things? Well, the squiggly things are actually the DNA. So in this case, what's happened is the nucleus of the cell, the DNA has supercoiled itself and is now, in the case of this one, going through anaphase. Okay, so it's being pulled apart. Okay, so we really don't have a good example of the other phases. I mean, we've got anaphase, we've got metaphase here. This is more than likely prophase or prometaphase. Okay, you start to see that there's these separations, which is really, really cool, of the DNA, and that's where you're now getting mitosis. So since mitosis is occurring, which part are we in? Oh wait, we're going to be in the zone of cell division. Okay, so this is zoomed into the zone of cell division. Look at where that's at on a real root. Note we have the root cap down here on this end. Okay. Okay. All right, so now we're going to look at a cross section of zeomase. Zeomase is a monocot, and this monocot root, we're going to zoom in and start to label structures. So here you can see that phloem runs around in a ring. Okay, this ring is generally about right here. Okay, then in the middle of this, you're going to have a region called the pith, and then you're going to have your xylem. Okay, your xylem is going to carry something specific. What is your xylem actually going to carry? And which direction is it going to carry it? So in this case, you've got your xylem, which is actually going to carry your water and nutrients up the tree. And your phloem allows things to flow back down. That's an easy way to remember it. Okay, well then you've got now this endodermis layer right here. Alright, and then you've got a cortex. The cortex is going to go from the endodermis out to the epidermis. Excuse me, that line was a little too long. Epidermis is here, and cortex runs from here to here. Okay, let's zoom in a little further. So that way you can see phloem tends to be bundled and xylem tends to be these large open cells because it's moving a lot of water. Well, how does water move up? Well, when you've got water in the leaf, okay, you have an opening that allows it out, which we'll talk about here in a minute, called a stomata. And water likes to hold on to each other because they have polar ends. And so as one water molecule leaves and jumps out of the leaf, another one gets pulled up. And then you also have water that wants to cling to the inside of the xylem. Okay? It wants to cling to the inside of that xylem, and it will actually pull itself up through something called the capillary action. Alright, let's look at a Eudicot root. Remember, like I was talking about earlier, Eudicot roots, you're going to see this X pattern. Okay? You also have your endodermis, which is going to be around here. You have your cortex, which is going to run from your endodermis out to your epidermis. Okay? Zoom in a little bit more, and you can see the xylem and the phloem. You also see cortex, and you can really see that endodermis layer. And this is zoomed in even further. Now you see the xylem is very, very open, and the phloem, you can see that there's actually stuff in there. Pretty cool, right? Alright, so now we're going to look at the shoot system. So, in this part of your lab, you're going to have to do some labeling, just like you've done previously. But there are some images that are not going to be in your lab manual, this being one of them. So in this case, when you take and look at the shoot tip, coleus, you start to see that there's these, well, pretty structures in my opinion. You've got this structure that comes out like this. That will eventually become a leaf, so that's called a leaf primordia. Then you've got these little buds right here. These little buds will eventually become branches, so you've got an axillary bud meristem. Okay, remember that we saw meristem earlier. Okay, so that means it's going to be a developing portion of the plant. Okay, this is just a zoomed in so that way you can see a little bit better the cells that are actually there. Alright, now we're going to look at the leaf cross section. So the leaf cross section, in this there are a lot of little things you're looking at, and in this case it's small cell layers. So you've got your upper epidermis, which is this single layer here. You've also got a lower epidermis down here. Okay, so upper and lower. So this is the top of the leaf, and this is the bottom. Why is that important? Well, there's a couple reasons why. For one, if you look at the leaves, and hopefully you see this when you go out and identify some plants, you'll see that one side tends to be darker than the other. And that tends to be something you see in Eudicots. So you've got one side that's darker than the other, and the darker side's usually facing towards the sun. So that darker side allows for more light absorption, so you're exposing more chlorophyll on that side. Now, we'll get into why that is here in a second, but just note that you see typically the top side of the leaf is darker. So on the bottom side of the leaf, it's not as dark, and it's also got these little structures, these little openings. Okay, that little opening, stomata. Okay, stoma. You have two guard cells. You have a guard cell here and a guard cell here. Those two cells are going to swell and contract, okay, to open and close this. So when they swell, they open up. That means the stomata is now open. If they swell, they're going to be pulling water into themselves, and they end up making two little C shapes, okay, to make a circle. Now, inside the leaf itself, this is where we're going to talk about, you've got two different layers. You've got this one layer that looks like a bunch of columns here. These columns, these remind me of a palace, okay? So that's the palisade mesophyll. You're trying to pack as many cells on this top side of the leaf as you can, so that way you can have as much photosynthesis as possible. So this is a huge advantage, packing this in like this. But what about the spongy mesophyll? Does it not actually also make sugars? Yeah, it does. It goes through the process of photosynthesis. It's also going to have chlorophyll. So both of these actually have chlorophyll. So the leaf itself is a photosynthesis machine. Okay? Alright. Let's zoom in a little bit more. You can see the difference here between the palisade mesophyll and the spongy mesophyll. Remember, like I was talking about, those columns, okay, and the palace. Then you've got these gaps, or these spaces. These spaces in the spongy mesophyll actually allow for gas exchange. This gas exchange is carbon dioxide and oxygen. It wants to intake carbon dioxide to be able to make the sugars, because the sugars, remember, they're C6H12O6, so it needs that C and it needs that O. Okay, and then it's going to take and have to actually give off oxygen as a byproduct. Okay, so note here that there's a layer you can see in the top right hand corner here, but it doesn't have cells in it. This is called your cuticle. It's also usually referred to as a waxy cuticle. Okay, that waxy cuticle helps shed water in the rain, and it also helps protect the cells from wind damage. Okay, so it has a dual function. Alright, oh wait, and xylem and phloem are still here. You can see that there's phloem on this lower side and xylem up here. Xylem and phloem in this case, says it's a leaf vein. So that leaf vein is allowing for water to be brought to the leaf. Why does the leaf need water? Well, for one, it's going to need it for the process of photosynthesis, but two, it also uses it to help in cooling, because if the leaf gets too hot, it will wilt or wither, and you end up losing function of the leaf. So you also have to let it absorb heat. Okay. All right. Now we look a little bit more at the stomata. You can see here the guard cells. They're very prominent, and notice that there's a flat opening here. Now remember that this is a cross section, so you're not going to be able to see this whole channel or this whole air gap. It looks like it ends here, but it really doesn't, because it's going to be open to other areas. So you have to kind of think three-dimensionally on this one. Okay. All right. And so this is what a stomata looks like. So as the water leaves the stomata, it's going to close. Okay. As the water fills in, it opens because those cells become what's called turgid. They swell. Okay. So in this case, you can see this one is fairly turgid. This one is kind of turgid. And this one, it started to actually close down. Okay. All right. So the next video we'll be looking at salinity. So please stay tuned.